Hey
We good?
everybody, welcome to the Growing with Fishes podcast. I'm Steve, and this is the Growing with Fishes podcast. We do this show every week, trying to educate everybody on sustainable cannabis growing. Um, we have Tommy with us.
Hey there, everybody. Glad to be back this week. Got some pretty exciting things we're working on. I'm going to be taking a trip down to Del Mar, see my sweetheart, which is always exciting for me. And but I'm going to be on the show here, and hopefully I don't know if I'll catch everybody next week. I might be on the road, but glad we're here, and I know we got an exciting show today.
We got Roger.
How y'all doing? I'm Roger. I run iloveggrowingmarijuana.com, and it's good to be here tonight as usual. And we got a great guest for y'all tonight. You're going to really enjoy it.
We got Mr. Green Jeans.
Try to keep up, you guys.
Hey, what's up, everybody? Mr. Green Jeans, the original, except no substitutes. Really excited to be here. This is a great crew.
We got Fish Ganja Guy.
Yeah.
We got Brain Grow.
Evening, everybody. Welcome to tonight's show.
We got Dutch Blooms.
Hey guys, stoked to be on again, and super stoked to hear from Dr. Efren.
Hey, Josh.
What's up, buddy?
And then we have Dr. Efren with us this week. He's a mycorrhizal expert, and I had a chance to listen to his talk when I was up at the Science of Regenerative Organic Cannabis Conference up in Portland. And he's going to be at the along with me and Josh up at the one here in Humboldt on what's the exact dates, Josh?
The exact dates for the conference are the 16th through the 18th, and then we're doing a microscope class with Dr. Elaine Ingham, on the 14th and 15th. So big week for us next week.
Oh yeah. And so thanks a lot, Dr. Efron, for coming on. Why don't you tell everybody a little bit about what you do and what mycorrhizae are?
Oh, boy. Yeah, it was good to meet you there, Steve. Thank you for the invitation to be here. Well, it's a big question. The first thing that comes to mind is that we tend to use the term mycorrhizae pretty loosely, but I keep thinking of my major professor, that keeps correcting us. And so he says, okay, so you don't sell mycorrhizae because mycorrhizae by definition is the association between a fungal that creates this symbiosis, this mutualism with the plant roots. And so mycorrhizae is the actual association, is the bridge, is that structure that is interface with the plant, between the fungus and the plant. And so what we actually place in our substrates is mycorrhizal fungal spores. So spore is sort of like an extension of the fungi, the reproductive structure. And so we apply mycorrhizal fungal spores or mycorrhizal fungi. After that spore germinates and creates a tube, which is called a hyphae, and then mycelia. And that mycelia, to actually keep going and growing, needs sugars, needs energy, so they connect to the roots so they can get the sugars from the roots, which are formed by the plant. So that's kind of a quick definition of what mycorrhizae is and what mycorrhizal fungi is. Not all fungi can do that, as I say in my talk. Any fungi, we can place them in one of the three categories, and no matter what the fungi are, One is the decomposers, which are very well known, and we usually probably see them and step on them and eat them and touch them pretty much every day. Those are the ones that decompose organic matter already formed by the plant most of the time. And then you got the parasites that they go after living tissues and causing diseases. Those are the pathogens. And the rest of them will be the mycorrhizae fungi, the ones that need the energy from the living plant. And there's a benefit between them. The plant gets nutrients that they cannot absorb by themselves, and the fungus gets the carbohydrates that they cannot make. That's why they're not plants. They're not photosynthetic. And so any fungi, you can place them in those three categories. Most of the ones you find on compost teas are decomposers, and breaking down organic matter. So mycoroots, I started back in 2002, and basically it was sort of like working at the Oregon State University and the forest science department. I was doing a lot of mycorrhizae assessments for research for my own, and fungal identification, forest fungi and such. But I can tell, I was involved with the, helping a couple companies to start their business because I was just, friends of them. And, they started sending me stuff to me because there was no one that could do assessments, colonization. Does the fungus actually working? What we applying is actually working. Because one thing is you apply it and you apply all kinds of different things, substrates, nutrients, teas, and all kinds of stuff, bacteria. And then you say, well, it's working. But if we stop there for a second, it says, well, how do you know which one is the one that's working? Was it the bacteria or was it the substrate? Whatever you did, what did you measure that it gave you the answer that you are blaming for the effect? And so that's the key part, because we want to try as many things all at once, and we don't have a control, or we don't set up control, or we measure the wrong parameter on the plant. And so it's a little loose. And from the science background, you want to be careful because people will follow you. If you got a strong followers, and then they follow you, and they did that. But science is pretty objective. Does it or does it not work? Did I measure the right thing? And so based on that, people wanted to test us. So I started getting a bunch of samples and work, and I would do it out on the side and then do it at home. And pretty soon, I was making more money doing that than actually doing research at the university. And I said, well, maybe this is a business. And science funding was getting drier and drier, and with all this administration back in the Bush area, the pretty much the funding starting to reduce. And I said, I might be able to do something along these lines, out of the comfort of my home. And so I start doing that, and just word of mouth, I keep getting more work and more work and more work. And I said, well, I guess this is my niche. I love doing this. The mycorrhizae thing, I learned it when I was doing my dissertation. And it was basically, I had to assess mycorrhizae colonization on pretty much any plant, on a glacier forefront in the Washington Cascades. So I could determine, arbuscular mycorrhizae, ectomycorrhizae, ericoid mycorrhizae. And so I said, well, I guess I can detect all these kinds, and so that would be the core of my work. And I was a little diverse. I was doing mycology studies, small mammals. They eat mushrooms, and usually for science, there's no clients in the industry, to look at poops, under the microscope and see if they eat fungus or truffles, which I did that for a few scientists at that time. And I was doing fungal identification, mainly ectomycorrhizal fungi, for the Forest Service, and I'm still doing it. One of my specialties early on was truffles. So, so I work with truffles from Mexico, and I travel a few countries in the world exploring forests and collecting truffles. So mostly diversity, and not just the edible ones, but any truffle, any below ground fungus that grows underground associated with trees. And so, here at Oregon State, we had the world expert on truffles in the world, Jim Trappy. And so he was my major professor. And so we travel many places, collecting truffles and having fun because, work was fun. And so, and he was a pioneer in the mycorrhizae research along with other people. And I had the privilege that Oregon State in those days, in the 80s and 90s, was kind of the mecca of mycorrhizae research. And because all the scientists that were there, at some point, Corvallis had about 60 mycologists in town. And I did that count because one time we went to the bar after 5, and I said, they call this symposia, and all of a sudden, pretty much every table where mycologists is just talking fungus, fungi. And I started counting them, and I said, "Holy moly! How can you ever put 60 people living in one town that work with fungi?" From microscopic fungi to mycorrhizal fungi, pathologists, and so on, and we know each other, and so with that, with because of that, we get a lot of scientists visiting town, stopping at the lab, and so you get to meet a lot of these guys. They're doing all this research all over the world. From Sweden, England, Australia. I mean, Latin America, you name it, everybody was stopping by here and working and volunteering and cooperating, writing papers and stuff. And so, and then unfortunately the lab disappeared. The funding got shut down, people retire. And luckily by that time, it was already 2007. So mycoroots was already established, like, okay, well, I guess I'm on my own. And if I can make it in 2000, if I made it in 2008, which I didn't know at that time, I think if you remember, we hit pretty bad economy and the recession part in 2008. And at the end of the year, I said, "Wow, I made it." So I guess this is a sign that I can. If I made it in 2008, I can live on this thing. And you know, one of my early clients was Soil Food Web, because they were established here with Elaine Ingham, and so she contacted me, or we connected somehow, and I started doing mycorrhizae assessments for them, and so there was one of my early clients, and then she sold the business, and I'm still doing that for Earthforth, and so. And then I get my own clients from all kinds of directions. I don't know if I can name them, but I got some pretty good big names from the NFL. They're doing sports, the fields, the training fields or their fields, um, evaluating the mycorrhizae fungi on their turf. You know, what are they doing and how do they? What's their status? Um, and then I got associated with a guy from New York that does sustainable urban landscape for the New York Central Park. And so he started asking me stuff, sending me stuff, and he liked my work, and I like to get stuff done quickly because the quickly you get your job done, the quickly you get paid. I'm not on a salary, I'm not like a steady income, so which is very risky in this business. You probably all know that. Paychecks don't come on the 15th or the 30th.
What's your paycheck? I got a checkbook. I write checks, but I don't ever get a paycheck.
Yeah, I've started getting
come on the 50 to the 30s if they come at all.
Or a day off.
Exactly. So I work Saturdays.
You want to be a farmer, you better know you never get a day off.
I work Saturdays, Sundays, or evenings. So this guy started inviting me to big projects like the St. Louis State Park. They did a big renovation in St. Louis, and so they apply mycorrhizae. There.
Are we back?
I think we're back.
Oh, okay, so we lost Dr. Efren. It wasn't me. I thought I might have froze up. Okay.
Yeah, we lost brain growth too.
He's coming back right now.
Yeah, so we're doing
good thing about Hangouts. It does function very well when you have to get back in. You just click a button.
Yeah, we're doing the Expedia.com in Seattle. Turning over 20 acres of soil and following mycorrhizae fungal path. Because and also the Obama Library in Chicago is another one we're doing. And I don't know what else. They just keep coming, at least 2 or 3 a year, new ones that I just don't know. So I have to Austin, Texas. So I'm all over the place. Which brings to the point, and I let somebody else ask a question, brings the point to me. Are you still guys there? I think
yes,
okay.
I'm still here.
To me, mycorrhizal fungi assessment, evaluating them and their roots.
We're fine.
It gives you a point that a lot of good things are happening in your soil or in your system. By the time you find a mycorrhizal fungal colonization thriving in your roots, it had to go through a lot of things, a lot of filters, this temperature, substrate, pH, nutrients. And so it's sort of like a climatic success in your root system. And so if you see them at a nice level, it kind of gives you an assurance that your plant is doing well. Especially if you inoculate them. If you did inoculate them well, don't expect to see much unless there were spores in that substrate by chance. And so that's what I follow the path, because most of the plants in the world are mycorrhizal associates. And so it gives you something to grab on, to actually detect. And that's the difference with bacteria, because bacteria, they're not easy to detect in the root system if they go inside, unless they're nitrogen fixing bacteria where they create nodules. But if you apply free living bacteria, there's no signs in the roots that you can see, oh yeah, they did their work. That's not how they work. It's either they're in big numbers in the substrate, but there's no actual structure that you can measure in the roots. So it's all cause effect that you can see if you got the right controls. But otherwise, there's no other way. With the mycorrhizal fungi, you can actually see them inside the root system. And there's question about, well, who said that 1% is better than 30%? Well, that's all we got. And if we go even further, we can question one species of mycorrhizal fungi can colonize 10%, and another species B can colonize at a 40%. But they're both efficient. And so what does that mean? That this species A can be just as efficient at a low colonization level than B at a high colonization level. We can raise that question, which is very valid, but we have to determine what did you measure to see that, to say that. So that's an open question. And as I said on my talk, when there's graphs and things where they show arrows, this little organism influenced this organism, and the soil food web, arrows everywhere. That's very simplistic view. In reality, there's thousands or millions of arrows going everywhere at every second or minute in time and space, because the ecosystems are so dynamic. The only thing we know is that temperature slows down those arrows or those interactions as far as we know, but the ecosystem still's alive. And so if we extrapolate some of this knowledge to a reductionist way of growing things, which is basically, like when you grow vegetables in a small scale, or cannabis, it's a reductionist way of doing things, because cannabis in the wild doesn't grow with all these nutrients. It's an artificial environment that we're doing. And so since it is artificial, we're trying to find the holy grail, to mimic how this plant can grow more than expected. Because in reality, if you put a wild plant, cannabis doesn't know that it needs to produce a lot of THC or CBDs or beautiful flowers in 90 days. And so we're asking this plant to do magic, to do miracles to our benefit, because it's an economic, thing that we're trying to do here. And so we're always finding what is going to work the best for my strain. Does it work with every strain? It's always that question. Does every strain has the same needs as the others? I don't know. I don't know anything about cannabis. I had no idea how to grow cannabis, but I can raise some questions. As how does, someone was mentioned lactobacillus. Okay, you got one strain of lactobacillus, and keep that in mind. Does that strain work the same for every strain of cannabis? Does anybody knows that answer? Has it even been tested? And what did actually you measure to determine that question? And so that's just one thing. And if you're starting to pile up the dozens of bacterial species that people apply, dozens of mycorrhizal fungi do you apply, are those strains that you use in your inocula, are they the same in every package that you buy that every company produces? No, they come from different generations. Maybe one common mycorrhizal fungi out there is Glomus interradicis. Now it's called Rhizophagus interradicis. There are a lot of strains out there. Not all the strains are the same. Maybe one was isolated from citrus, the original strain, but others came from potatoes, from bananas, from peppers. Who knows from where? From corn. And so they carry the same name, but do they carry the same physiological traits every generation? Because they're cloning them. And so when does a clone loses their physiological traits or powers? We don't know. And so it leaves a lot of questions, you know, science based, that it just makes you, and not skeptical, but the message I like to give to people is that there's no rules. You know, uh, things change. You know, the light, the watering, the type of water, the water quality, when do you water it, and on and on and on and on. And so it creates a very factorial experiments. And so we do the best we can. Uh, and in science, when you do experiments, if you don't have the right controls or measure the right parameters, then you may producing, I don't want to call it fake science, but not, there may not be consistent results. And so
I'll call it fake science. No, claim your data, dude. That's
yeah,
why
I mean,
you have it. That's why you have paper on the wall. Speak it true. I mean, because like I watch people in their grows, they have one grow and then they change 4 parameters for the next grow, and I'm like, dude, how do you know what you did? You got to change 1 parameter.
and the thing is that most growers are not for science. They're to grow, to grow to get profit. Along the way, they find formulas or different things to do things to get things better, but they don't have time to do science because they're on a paycheck, or not paycheck, but profit, and and so I'm wondering, and I think I heard there's a cannabis research institute in Michigan or some university where actually doing all kinds of things, but I don't know what kind of a research they're doing, or they have time, to actually test all these things, because
If it's Mississippi, they're really doing a great job.
yeah, is that Josh?
Yeah, I'm being
So
sarcastic.
no, but I mean, it brings the mind to Josh because Leighton, I don't know if I should mention his name, but Josh,
Yeah, no, go ahead, talk about it.
Josh is part of the what we're trying to do, and again, this may not be a strict science, but we're trying to pinpoint something very basic, and then it's along these lines. So the question was, here's a cocktail of species of mycorrhizal fungal species. They're sold. And the question that came up was by Leighton, says, yes, but what species is they actually working? Basic question. Is it a synergism between the 5 of them, or is it one each? But which one is it? Is 1, 2, 3, 4, 5, or the combination of all of them? And so he convinced the producer to give them separate species instead of the cocktail. And so we apply it individually, or Josh applied them. I'll give all the credit to Josh. I'm just the sort of like the do it this way, do it that way, but so the credit is to Josh and Leighton. And so they're going to grow them separately and to see which one does best. But again, we weren't much concerned at this point of the plants got better or bigger. We're more about, did they actually colonize? And so, unfortunately, the plant had to be harvested earlier, and we didn't give the chance to the mycorrhizae fungi to actually get in there and get established at enough time to do much.
And that's a good point. I'm just gonna, how long is that? That's like 90 days, is what you said.
Yeah. I like 90 days to 120 days, but a lot of times it's not possible because you need to harvest, and so. But we had signs of 2 or 3 species, if I recall, that show colonization. And I said, "Whoa, that's a good start." And not just because of that. Here's the kick. The kick was that the substrates were high in phosphorus, really high, which is that's what people do, because I'm assuming cannabis like high phosphorus. And so the paradigm is that most mycorrhizal fungi do not like high phosphorus substrates or soils. They tend to reduce their populations because they're not needed. That's what mycorrhizal fungi do. They secrete enzymes to break phosphorus down to simple forms that they can uptake them, in the solution, and be taken by the plant. And so when there's high levels of phosphorus, the fungus is not needed. The plant is taking it as is, and so that's the paradigm. And so when we found colonization a little bit on high phosphorus, I mean, I'm talking high. The rule of thumb is 20 to 50 parts per million of phosphorus in the substrate, because that's when the fungus actually works. It sucks it all in when they're very little, and it's more efficient. And so I said, "Whoa, this is a good step. This is a good sign that there are strains that may be able to live on their high phosphorus content in the substrate." And this can be a breakthrough because, first of all, it breaks all the paradigms. Okay, there are strains that can actually grow. It's the matter of finding them. And I didn't know at first, this is, that the substrate was high in phosphorus. It just, let's see what happens. Because if I knew naturally or instinctively, I would say, well, there's no point of growing them if there's high phosphorus in the substrate. Let's not waste the time. But I'm glad they didn't tell me that, because they did it anyway. And so when I saw the results, I said, oh boy, this is a good thing. And so now we're trying to set up a bigger experiment, maybe a longer time, to apply those, species, maybe all of them again, or just selected ones, and replicate this thing and have a nice control. And
what would be the result of that? Would you have, they would tolerate a very high level of phosphorus, and you'd have super
that's
phosphorus soil?
that's what we think. And if that's the case, and then we can select that species or that strain. This is okay. Why don't we test this one? I mean, there are all kinds of ideas we can come up with after this. Okay, this strain from this producer is we're gonna market somehow. This is okay, this strain. Why don't we compare the same species from a strain coming from a different source and test them both? And if one does better than the other one, I think we're getting closer to the answer. Yes, this strain does better than that strain, even though they're the same species at a high phosphorus levels in the substrate. Under the cannabis strain that Josh is using, we cannot say that will work for all the strains, see? Because it's very specific to our question. You know, we got to say, okay, what strain did you use, Josh? And now the question is, would it work on another strain of cannabis? Let's try, and all of a sudden you need a bigger greenhouse and bigger this and bigger that. And Josh may not be able to do it because he needs to make money.
And it might not just be the phosphor. There's
It could be other stuff, but we're trying
like
to.
we might be focusing on phosphor, just like for the longest time we forgot about CBD in the plant because we were breeding to grow indoors clandestine, and we bred out the CBD without recognizing how valuable it is. And here we can find ourselves in a situation similarly, where yes, we're aware of phosphor, but this colony, this dance of life that we're trying to create in the soil can be turning on 6 other switches that we don't even know to ask the questions for yet.
Well, let's not forget that also, if it's a high concentration of phosphorus, there's a possibility that other minerals are being locked out or on the verge of being locked out too, on the other side of the spectrum of availability. When you come, we're talking about making the molecules and the minerals available to the plant by breaking it down with the mycorrhizae or the fungi or whatever. Yes.
Yeah. So it's kind of exciting, when you sit around and start talking about all this, interesting questions that they can be tested. And but again, I'm glad Josh is available to do this, because, well, if we were to do it the right way, obviously it will require, nice funding, greenhouses, spare time, consulting time, and all the money that goes along to set up this thing. That could be huge because you need a lot of replication and somebody to watch the plants and take care of all this stuff. It's not cheap. It's not cheap. That's all I can say. And most growers do not have time for this because they need to grow their crops for profit. And research institutes, if whoever is the head of the research and it doesn't have these kinds of questions, or they're not priority, they'll be put aside, but you can set up the same questions for pretty much anything that you apply to, as nutrient levels, bacterias, but I mean, again, that's the issue with the cocktails of bacterias. The same thing. They sell you a dozen types of bacteria in it at different levels, different colony forming units. And when you apply them, the question is, which one is doing the benefit? Or what benefit? What are we going to measure?
Take the same colony and add it to a couple different plants and a couple different substrates, and just because you have everything identified in the colony that you're adding doesn't mean they're all going to take up in the media the same way. And one plant is going to give better sugars and another plant not.
And that's where replication comes into the picture, and obviously science-based, you got to bring a statistical analysis, set up your hypotheses well, and before you even do anything, set up your experiment design. So when you're done, you know what you're going to measure, at what times, the same way every time, so it's all uniform. And at the end, you're going to analyze those measurements and say, yes, this is significant based on the sample number, which brings another question or very typical problem. And this goes for a lot of commercial people. and the people that sell stuff or sell your products, clients wants to believe what they're selling you, and they tell you, well, we did all these trials and all this stuff, but the basic question, I tell people, my clients, that ask them immediately when they show you graphs and things, ask them immediately, what is the sample number? Because if they're basing their data on one sample or two samples, you gotta be kidding me. That's not gonna fly. I mean, what is the sample number? And if you throw
have a null hypothesis with one sample.
Exactly. And before you ask for the sample number, ask them, what is the hypothesis? And they're like, what is hypothesis?
Yeah.
Like, that means they're not prepared. And it says, what's the sample number? What's the N on a statistical analysis? What's the N? What's N equals what? Don't tell me it's 2, because that leaves you nothing. And what is the control? And what time of the year did you test them? Was it indoor, outside, the lights, I mean, you name them and they're like, whoa, they start pedaling back. Like, they're not prepared.
That's an excellent point. Yeah, such a great point.
Because variation, you mentioned it, variation is always going to happen. And that's why statistical analysis, it's key to actually detect those little differences. When we can't, I mean, you set up an experiment, only 2 factors, one thing and the control. That your question is, does they get bigger? That's all you're gonna know. That's all you care. Okay, that's a basic question. Are the plants bigger than the control? Okay, maybe you don't need to measure anything. And if the control are stunted, is short, and the other treated were like 2 meters tall, you don't need a statistical analysis for that. The statistical analysis, in my point of view, is when the differences are subtle. When the measurements are so hidden in between the variation between the individuals that you measure, when the numbers are all over the place, when maybe there's some outliers, and that's when you start to say, okay, I need some statistical analysis so I can say that it was a statistical, but it's not blown away statistical analysis, so it's some basic stuff, and I like my to educate my clients too. And actually, I learned this, on my PhD, because when you learn a PhD, it's not going to be it's not about how much do you know. It's more about, do you know how to ask good questions? Because when you go to a PhD, they grill you. They make you go to the floor and say, forgive me, please be nice to me. I don't know nothing. They bring you to make you humble, because we don't know nothing. We know so very little. We know a little bit of our topic, but after that, the main thing is learn how to ask good questions regardless of the topic. And not so much about skeptical, but just ask good questions, because the more good questions you ask, the more those brains move. And if those brains are attached to the scientific method, and it says, be careful with the statements you make, because someone with the science background is gonna stop you, cold. And like,
Don't ask a question you don't know the answer to.
Experiments is that I'm not accredited. I help put my wife through school and get her OBGYN stuff, and I come from a science family, but I don't have paper on the wall myself. But what I do know from hanging around all these really smart people is that I acknowledge that I have been wrong about way more in my life than I've ever been right about. So when I'm approaching experimentation, I'm just assuming I'm wrong. And unless I can double, triple prove it, I'm still going to assume that I'm wrong. And a lot of people don't think like that, especially sales folks and whatever.
Yeah.
They think it's the one thing, and it cannot even be what they're saying, and they don't set up the experiments to prove themselves wrong. They set stuff up to make it look better for themselves, and they throw out bad data, and that's not what the team is
I think it's important to establish a benchmark, which we were dancing all around, but you have to have a benchmark.
And that's exactly what I was going to say. You're right. You touched that point. Both of you touched that point, because it's nothing wrong. It's not like, I'm not saying if you don't follow the experimental design, I don't believe you. No, that's not what I'm saying. If you said, I did this, I did that, it may not be a scientific experiment, but I did this, this, and this, and I got these results. Based on what I did, this is what I found.
And then I changed this.
There's some weaknesses. There's some weaknesses on my conclusions, but this is what I found out. What did I learn from this? That's a great point. When you say, I only had 2 samples. Okay. It's honest way to say, I only do 2 samples. Okay. But those 2 samples could mean a lot for the next step. But it's not a decisive conclusion. As long as you're being honest on what your results or your conclusions are based on, people will have all the information they have in their hands.
Which is, I mean, sorry, just to cut in, that's like the stage we're at, where we did like a loose test.
Yes.
And now we're going to do something a little bit more educated.
Yes.
And then if that like pushes us forward, then we'll take it forward to a real scientific test.
Exactly. And it's very likely if we get peer review on this, somebody will say, well, it would be nice if you did this. Of course we do, but we're limited, for funding, space, time, and all that. But as long as you're upfront on how you set your conclusions, it's all good, But don't tell me that you don't know your sample number. That means you have no clue what you're saying. or you're lying. Said my sample number was 20, but it wasn't true.
So you guys are basically at the point where you've built a foundation to build on and to go forward with your experimentation, because you provide, now you built a benchmark at least.
Yes, usually science starts with speculation.
Right. Yeah, we're going to give it a shot.
In this case, it was Leighton pushed the deal.
Yeah, and Leighton said, I got this, and I said, Leighton, you got a lot of questions, my friend. And let's start one step at a time. And I'm glad, persistence, push. And it's very likely that, this may grow and grow and grow. But, this is okay. What is the species? I mean, if you guys were sneaky already, you're probably thinking, so tell me what's the species? Regardless of the strain. Because that's the key, it's interesting.
I don't even know. I have no clue. I just
my question.
I know the numbers.
You said a while back, and then my internet cut off, so I don't know what happened. You were going to say, well, we're going to let you have a question, and one of my questions was going to be, so how does one get into creating their own strain of fungal mycorrhizae?
Well, that's a good one. That's something that is one of my, what you would call it, dreams. But there's a collection. There's an institute in West Virginia that holds pretty much all the strain, not all strains, but a lot of species of mycorrhizae fungi, and therefore research, commercial, and so on. They sell you, enough spores and so on. The key is, well, I'm going to tell you the secret.
That's what I was hoping for.
I mean, the secret of the whole thing is ecological thinking.
Right.
So let's say you're looking for a species that is mycorrhizae fungi that is, well, let's take back. You're looking for a plant species that is salt tolerant.
Oh, cool.
In the world. Where'd you go and look for it?
Mediterranean.
Or marsh.
Well, yeah, you go to the Caribbean, some beach.
Right.
And so you start with there's no plants at all. You start walking from the water towards the land, okay?
Yeah.
So once you start seeing plants,
LOL.
what does that tells you? That plant is subject to salt water. Most of
You
the
probably
time.
have no idea how happy you're making Roger right now.
Yeah, because I want to grow pistachios, salt water. Yeah.
Okay, so you says, okay, this plant is likely, is obviously in our front of our eyes tolerant to salt. Now, let's go down below the ground. The question is this plant associated to mycorrhizae fungi or anything else, bacteria or whatever? And so you pull some plants, some individuals, you do the clearing and staining, or you send them to me, and I looked at them, and I saw, and I say, no, there's no condensation, and I'll keep my secret. I won't tell you what it is. No, if I tell you, yes, they're colonized with mycorrhizae fungi, then right there you have mycorrhizae fungi that is tolerant to salt levels or high salt levels. So you test the salinity on the soil and confirms that yes, this soil, their substrate is high in salts. And if you got fungus in there, that means this fungus is tolerant to salt. Let's take that fungus out and see if we can isolate it and propagate it and have a strain. We don't know the species name yet, that is tolerant to salts. And that's how you start. And then you send them for identification or molecular or whatever. Now you have a strain that is tolerant to salts. So you do the same thing with any particular trait that you're looking for. What about drought? Tolerance to drought. Where would you go and find that? You go to the Mojave Desert or some very dry ecosystem and start looking for plants that they somehow survive.
So would you then harvest that fungi and propagate it and test it against whatever, isolate it and test it against whatever you're trying to grow?
Yeah, it probably would take a couple of years of isolation and testing and recolonizing it, and the thing is that I'm sure it's not going to be only one thing. I'm sure there's going to be quite a few things, not just one species. Usually you find a collection of stuff, and so based on that, and that's kind of what I want to do. What I want to do as mycoroots. Start isolating strains, not just more, not only consulting or doing assessments, but also isolating and propagating for specific projects, and usually they're high-end projects that are interesting on their native stuff. You know, they don't want to bring commercial stuff; they want to bring native stuff. And so I think this is one of the new lines of stuff mycorrhizae is going to be doing in the next few years. You know, trying to get local stuff. You know, vineyards. Vineyards is another high-end crop. You know, that might be interested. You know, they want their local fungi.
Well, can I
Where
just
where would you go? Excuse me. Where would you go if you want to find mycorrhizae fungi that are adapted to cannabis, that are already been growing with cannabis for thousands of years?
Equatorial regions.
Afghanistan.
Yeah, that too.
Humboldt County.
Well, will you go for where cannabis
Thailand.
plant? You go where cannabis plants, you know,
Grow natural.
and
Where they're
hopefully,
indigenous. In other words, where they're indigenous.
yeah. And so basically, you'll go to those areas, and hopefully, you get some strains that been growing with cannabis for a while, you know, and get them out, select them, and see if they perform well and high phosphorus, because you're going to be growing them artificially. You know.
Well, now would you not have an issue with, if you went somewhere like that, bringing those plant species back to the United States?
That's a good
If
question.
it does, go get them, but
It's always a question. But the commercial inoculant industry is already spreading species all over the world for many years, and luckily, these are not pathogens. Obviously, I would be more careful bringing other stuff, not the mycorrhizae fungi, but other stuff in the soil that may, like pathogens or such. So, in other words, if I was to do that, I would probably try to do the trapping over there and only bring, certain things after a growth or two. So you're bringing clean things, or a lot cleaner.
that's what I was thinking. So you need it all cleaned up. Yeah, you want to clean it up, not have any kind of the soil where you get those pathogens for the soil.
Yeah, or maybe just extract the spores and bring spore concentrates instead of just bringing all the soil. I mean, yeah, you have to think about it a little bit.
So you'd have to be there a while. Yeah, you just don't go and take a trip and for the weekend. You'd have to go and establish.
And I probably let somebody else do the trip. I probably won't go there.
Well, the portion I was gonna just try to, direct back to is the 90-day inoculation, right?
Yeah.
And like the way 90% of us grow cannabis, that's just not a possible system.
Yes, exactly. I heard.
Unless you're growing in an indigenous soil or in a living bed or a no-till bed. I mean, Steve, catch me if I'm wrong, but even the way you grow aquaponically with dual root zone, it's a cycle. It's not 90 days within the soil to actually colonize.
Yeah, we don't. Well, what we do is we do take that soil and reuse it. We are experimenting currently with doing some no-till stuff. And then I guess on that note, do you want to talk a little bit about that, Dr. Efren, about no-till and the whole, you educated me a lot the one night on how the mycorrhizae pull the nutrients to the plants and why it's, you would want to leave the root systems in and not want to take them away. Do you want to talk a little bit on that?
I'd like to say real quick, I'm actually doing that right now. I'm trying it out right now. Same thing, and then I yield. Yeah.
Well, let me know, Roger. I'd be happy to help out in any way.
Yeah. I'll get your information, man. I'll talk to you about it later. I guess Steve wants the doctor to answer this question, so I just wanted to throw that out that I'm trying that.
So what's the question?
Can you tell us a little bit about no-till and leaving the old roots behind on the, and how the mycorrhizal networks help attract nutrients, especially in outdoor soil?
You guys want all the secrets. So I gave a talk in Edmonton, Canada, last December, and the whole conference was basically no-till, and I was amazed how many people are pushing for the no-till agriculture, at a big scale, especially in Canada, um, and I think for no-till, from the mycorrhizal point of view, it's the best thing you could do, because you're leaving some root systems or the below ground ecosystem to keep it going, or not being destroyed. You're slowing it down. And so once colonization of the native community starts populating all those root systems, and you harvest your crop and you do very little damage, you're just leaving the whole system below ground pretty much, not intact, but it's still there. And so when you seed it again, the mycorrhizal system is still going, and it's just ready to kick in again. So you don't have to go through this spore cycle where the spores have to be dormant and it takes a while to germinate them and colonize. And so from that point of view, the no-till is a perfect system to maintain the below ground community ready to go when it's needed, as opposed to destroy them and let them start all over again, every time you rip the soil. One thing that I told them is, some of this no-till, they do cover crops, and to me, if they have a choice of selecting a cover crop, my suggestion was to use a cover crop that will be a mycorrhizal host. So because that mycorrhizal host will keep the below ground mycorrhizal fungi active and alive and propagating while you're taking a nap, it was taking a winter, and so they'll be ready to go and you don't have to re-inoculate again, in theory, as long as you check to see if things are kicking. But so that's another ecological observation, that if we think below, I mean, we always think, unfortunately, we always think about what's the above ground community. We rarely think of what could be happening below ground. We ignore that the soil is not just soil. It's the home for a lot of little creatures that do their job when you're taking a vacation, or or hibernating. So these guys will do all the nutrient cycling uh, for us. Instead of relying and fertilizing every year the same thing, and locking things up in the soil when they're not being used, like phosphorus. Um, so I think the no-till is a great thing. Um, the thing I don't know much about is the the same thing could serve to promote beneficial organisms, but it could promote pathogens too. So I don't know what kind of system those people have, and I noticed, or at least at this conference, I told them that if they can avoid winter wheat as a cover crop, I would suggest not to put winter wheat as a cover crop because, and this is my opinion, my experience, when I seen winter wheat roots, they asked me for condensation. I see all kinds of other fungi in them, and I call them dirty roots. They host a lot of other fungi that are not mycorrhizal fungi, especially uh,
So non-beneficial, non-beneficial.
yeah, they just I just don't like them.
Parasitic.
Yeah, I see a lot of
Parasitic.
opidium,
Oh, I like that.
opidiums and allomyces. All this fungi that have a part of their cycle is water. They have motile spores. They have zoospores. They're more related to animals. Some of their cycle, their spores have flagella, so they swim in the water, and so they colonize their roots heavily. And so basically, when you're done with the winter wheat, those roots are sort of like a fresh pathogens alive, that you're keeping them going. So if you can avoid winter wheat, do something, I don't know, clover or flax if you can. Some other plant, not brassicas, no cannolis, no mustard family, because those do not
What
host
about a
mycorrhizal
manure?
fungi.
What about a manure blend with the beans and field peas and all that stuff?
Well, that sounds good. You bring in nitrogen.
So
Yeah, because I've got
that,
no-till grow beds, but I'm sorry, Steve, but I've got a couple. It's kind of neat that this rolled around in the no-till because I tried it off a video I saw on YouTube 2 or 3 years ago, and it's kind of almost like your bed is almost like a compost every year. So you can just keep, you can work it still a little bit and amend it a little bit, but you got a nice bed there every year. But I'm sorry, Steve, go ahead, buddy.
I was I lost my train of thought. That's okay.
I was bringing it up to just talk about the time of colonization, and that essentially, unless you have a no-till system, you shouldn't even mess around with mycorrhizae. Is that what I'm understanding correctly, Efren?
Not necessarily. I mean, I think the no-till in this case, even though it was not to promote mycorrhizal, you're doing it indirectly. If your mycorrhizal plants are a host for fungi, mycorrhizal fungi.
So
I guess in a situation where we're using we're putting plants in a 8 to 10 week, 12-week cycle. We're planting them in a soil base, that's in there for a very limited time, 2 months, 3 months tops. Is there benefit to buying and adding that to our regimen given the limited time frame?
Well, yes. I mean, if there's no other choice, and I would suggest to put the inocula in the fridge for at least 2 weeks. It could be a month. It could be the whole time, because the spores are chlamydospores. They're resistant spores, and so they're basically survival spores. Once they're produced, thick walls, so they just stay in the ground. They don't germinate right away, and so the cold temperature kind of scarifies them. Breaks dormancy. What happened during the winter in the plant community, especially in the boreal zones. Naturally, they all mycorrhizal species. They're in the soil. Freezes, but that doesn't kill them. And
They just go
so,
dormant. Eh?
they just go dormant. And they come to spring, and so they went through a cold period naturally. And then spring comes, new root growth, fungi will wake up. And so you use that concept to do it to your commercial inocula.
On that note, what would be good companion plants to plant with cannabis to attract beneficial mycorrhizae? I know purslane and certain grasses have pretty deep and extensive ones. I think it was your talk that about grasses having up to 16 different type of associated mycorrhizae. I think it was. Do you want to talk a little bit on maybe some plants that might be good, and how some of them there isn't much science behind? Or
As I say, you guys want all the secrets.
That's what we're here for.
Let me send you a PayPal account.
You sound like us now. I'm actually building a network where you could have your own channel to get to do that PayPal and have the whole world sending you money through your PayPal. I'm working on it.
Here's the thing. So the companion plants, based on the concept, any composite, any plant on the Asteraceae family will be good. Now, that's just based on the mycorrhizal companionship. But I don't know what, and this can be tested. I don't know if there's competition for nutrients that may affect your cannabis plant. I don't know.
See, that was my thought when that question was being asked, is about, and the root zones being too invasive and robbing the roots of the cannabis plant. That's what I was thinking too, could possibly happen.
Yeah, because, maybe it's not, and I'm just thinking out loud, maybe it's not necessary to put a companion plant. Maybe, gosh, you guys.
I've got an idea. I'll throw it out and then you can nod if it's the right thing. You don't say it, okay? I was thinking, go ahead and take your big giant, like your bale of Pro Mix and inoculate it pre-grow, like be proactive and grow your, put your fungi in there and have it growing for 3 months while you're growing your grow, and then use that for your medium next time. Look, he's nodding. See?
Okay, shut that guy off. Put the camera off.
Okay. There you go.
Delete him from the panel. He knows too much. Yeah, that's exactly right. I mean, that's basically the no-till idea.
Right. So, like, what
Right.
we're doing in my garden is I'm inoculated, and
Yeah, so
when I start my new clones, I clone off my mother plants in an aeroponic cloner, and then I put them in a 4-inch mix, and I inoculate them with the same strain. This is where we're heading towards, right? With the same strain that is in my beds. And then when I put my plants then in the beds, they're already working on the same communication line, in quotation. They're reading the same language. Is that the right deal?
yeah, basically what you're doing is to get the mycorrhizae wake up and get active, and then you can top the plants, chop them, and you can probably leave the root system, or kind of smash them up or whatever, so increase the colony sources, and then you plant your crop, whatever crop you want to do. And so when you plant them, you're basically putting them on a very friendly environment that is all ready to go. And so
go ahead.
As the thing is that if you check that the plants that you put before your fancy crop colonize, and then you check for the spores numbers and so on, and then you have data and facts that it actually work, then you put your fancy crop, and then you let it grow, and then you check for colonization. Again, you have data that says what I did, it did work. If you don't do any of those checks, you're only assuming. I mean, that's basically it.
Sure.
I have
When
to
the,
say something in the grow that I've done where I use the same, and this is in a cloth pot, 5-gallon cloth pot grow. I took my clones and I put them into the last grow, same medium. I just basically I pull once it dry, I let it dry out. I snap off and kind of snap the root ball around and leave all the other roots except for the actual stem and the right there at the root ball at the beginning, and then I excavated it to be able to repot my plant, I go from 1-gallon nursery pots into the 5-gallon, and I put them in there. And guys, I'm going to tell you something right now. The hybrid, crazy, vigorous growth that I'm getting, I mean, there's shoots, they're coming out everywhere. It's to the point where we've been taking and pruning off the bottom of our plants and letting all the energy go to the top. I've got so much growth up and down the stems, I don't want to cut anything off. I think I might be doing this, but I'm going to tell you this. I also, something else about, like we were talking, it was mentioned about holding the nutrients and all. What I found is that this is something that y'all might want to keep in mind if you're going to try what we're talking about and reusing your pro mix or your mycorrhizae, your grow beds medium and all over and over like this with the fungi still active and cultured in there. But I think that you need to understand that it does hold nutrients. So peace. So it does hold nutrients. And so when you first go in there, you don't need to be giving it a bunch of nutrients either. My plants got super dark green. Really, and I said, right, and I got a couple spots, and I said right away, I said, we need to watch out, and there's so much in this medium or substrate or whatever you want to call it. So far that I decided to go with water, and my plants just went crazy. And they didn't stretch, well, of course, I went into veg period, photo period, but they didn't stretch a whole lot. They just went nuts. There were shoots coming out of every node everywhere, and it just stacked up, half an inch to an inch apart.
I have a question on that note. So, is there any difference between doing straight no-till and then taking your soil from? So, if you're doing pots, taking that soil from the pots and say composting them in between runs for 3 to 6 months and then doing that again, does that also maintain the mycorrhizal spores and fungi and mycorrhizae, or does that, and mycelium, or does that not transfer over? And is it better to do one than the other? Is there a big difference?
I had no idea, but I would think it would be okay. I think it depends on the compost concentration. And it's just a feeling. Scientists should not have a feeling. Should be objective. But people have asked me, can you put the spores, or I want to make a product with compost tea? I said, oh man, just the thought of having spores, they're big spores, and put in the compost tea to make a product and thinking, all right, I'm going to have compost tea and mycorrhizae fungal spores. Man, that compost tea is loaded with bacteria. And mycorrhizal fungal spores, they are vascular fungal, the glomus type. They're big, and they're basically a big balloon full of nutrients. And so you put bacteria and spores together, that seems like a party for like free pizza, like, wow, look at all this food. And I seen bacteria making holes through the cell wall, trying to get to those oils, those nutrients. And so I don't know, it's something to be tested to see if they actually, mycorrhizal fungal spores work with compost tea. Now, so if you have a high compost substrate, it's a similar thing, but less liquid, obviously. But I don't know how long with the spores can. I don't know if the spores can take such a heavy bacterial substrate for that long. But again, I'm just trying to compare to soil. Well, soil is loaded with bacteria too, so it's just a feeling that I'm guessing the compost tea is a lot higher uh, bacteria numbers than soil. That's just, I don't know.
I've also heard that overdosing your trichoderma can also heavily impact your fungi. Is that true?
Yeah, trichoderma is another beast. Trichoderma is a big decomposer fungus, and so I don't know. I mean, people in the commercial business try to give you all kinds of combinations and cocktails. Uh, you got trichodermas, bacterias, and this and that, and I don't know. I don't know if they actually test how viable those things are, when they put together after 6 months in a package.
That's what I
After
was thinking about,
after
shelf
a year,
life.
I don't know. I mean, not just shelf life, but what is
I mean,
what
the,
is
the fact that the reaction continues to grow inside a bottle. That's what I meant, actually.
yeah, especially if they're in liquid. Liquid form to me is kind of active, and the powder
Yeah,
form, I would think that things are still, sit
yeah.
still. But that doesn't mean that the bacteria cannot be on top of the surface of the spore. I don't know what they would do in a dry form. Did they do anything? Are they active or just waiting, to get in? I don't know.
Very interesting.
It's a
Yes.
lot of things. I mean, my question to those companies that sell those cocktails is those basic questions. You know, what does it happen on the package?
I think some of that.
Actively
Oh,
is this what kind of activity happens? Does the bacteria does anything to it, or it's just Latin? They're just waiting. Um, I don't know. Did they test viability? How often did they test it? Every
I think
month?
sometimes it's
Is
a matter of marketing. One company comes out with something like that, so every other company that because they're a commercial nutrient business, or would come out with something. I mean, well, no, we don't want to we don't want to mention any of the evil
that's so good.
nutrient companies out there. But go ahead, Steve. I just wanted to make that comment. I think some of what
Is there
Dr. Efren was saying was due with the fact that they just make a product to sell it, whether they
yep.
test it or
Is
because
there a
somebody else.
is there a certain anything that people can add to feed their mycorrhizae more? I know some people recommend uh, molasses or other certain things to try and encourage growth. Is there anything that
Well,
comes to mind on that regard?
I'm not sure how that worked because remember, mycorrhizae fungi are mycorrhizal fungi. They're not saprophytic. At least that's what we know. So trying to feed a mycorrhizal fungus with sugars already made, that means that the mycorrhizal fungus has some saprophytic capabilities, which I wouldn't be surprised because um, they can grow in vitro for a little bit of a period, and then they connect to the roots in vitro, because that's where they actually, want to get their nutrients. But I seen mycorrhizal fungal spores and colonization on the bottom leaves of turf, And so what are they doing? They're supposed to be in the roots, not on the little leaves. And I asked that question to other colleagues. I said, well, that's an interesting thing. I never seen that before. I said, I seen them. I seen them a few days ago. You have turf colonization. Obviously you're looking for roots, but sometimes you get the little piece of the bottom of the bottom leaves of the turf, Bermuda, very tiny leaf, and you see the fungal colonizing inside of the leaf. I said, these guys are breaking all the rules. And so um, there's a lot of things we take for for granted, or that we think we know everything. But the more we look at things under the microscope or below ground, we keep finding other stuff. I don't know if you guys have microscope, but man, once you start seeing things at a very, under the 40x or 100x, the whole world changes. Um, and I'm just going to throw this in, and I don't know how long we're going to be talking. I need my cookies and milk to go to bed.
Oh no, you can we'll wrap it up after this.
One thing, and this I say this to Leighton. This blows him away. I said, look, Leighton, the spores, the mycorrhizae fungal spores, and this is not new, and this is not my discovery. Inside of mycorrhizae fungal spores, mycorrhizae fungal spores have like 40 microns. It's like a balloon, 40 microns to 100 microns, 120 microns, full of oils and stuff. But there's endosymbiotic bacteria inside the spores that who knows what they're doing.
Does that mean sorry to interrupt, does that mean we can't see them with our eyes?
No, they have to have a special staining, a higher magnification, because all you see on this kind of scopes is just little things wiggling. That's as far as we're gonna see. So you gotta have a higher magnification. But the question is, what is what they're doing in there? Are they're probably transferred from the tube, from the hyphal, because mycorrhizae fungal spores, the arbuscular ones, don't have septa. So in theory, all the organelles and oils and nuclei are migrating back and forth. They potentially they have no septa, so they're not contained. The cytoplasm is flowing. And so when the spores are formed outside or inside the root, the hyphae swells up and makes the spore. It's just basically an extension of the hyphae, starting to swell up. The chemistry of the cell wall changes, make it thicker, and then this bacteria goes inside. They get inside, and if they're formed outside, well, the hyphae goes outside, and this bacteria goes inside where the spore is. What is what they do? What could they be doing? Trigger germination is one possibility. Cycling some of these nutrients that are inside. The spores is like a reserve of glycogen and sugars that are needed for energy when the fungal hyphae is going to germinate. It's sort of like the energy is stored there. And so when the spore germinates and creates new individual, the energy is coming from what's inside the spore. Once the fungal hyphae grows and grows, and if that fungal hyphae is not doing any saprophytically, it needs to connect to the root, because they need the energy from the root. If it doesn't find a root, it's going to die. And so it's sort of like you got one shot. If you're going to germinate, you better go and find a root, because if you don't find it, you're done, buddy. You cannot survive saprophytically for a very long time. It's very likely. And so that's why I say, if you check root colonization and find the structures inside the roots and determine a colonization level or just the presence, there's a lot of successes that have to happen for that fungus to be inside the root. And that's why we're using mycorrhizae colonization as a way of a sign of healthy soil.
I wanted to ask something earlier. When you were at the very beginning of the show, and I wanted to listen to you before I try to interrupt and ask a question, but you kind of hit right back around on it. So you would consider mycorrhizae fungi as synergistic. It brings all these different.
Well, we consider them mutualistic, but synergistic.
Okay.
It's very likely that they can, here's the question back to the Josh experiment.
Yeah.
If we combine the 5 species, we don't know if those 5 species do something they cannot do independently. If the 5 species work and colonize, unfortunately, we don't know which one is colonized, because once you look at inside, they're all going to be looking the same way. And so we don't know which species is the one actually growing. But those species might be doing something together, or they're doing something together with bacteria, or they're doing something, there's so many arrows. Remember the diagram? There's so many arrows that they could be interacting with each other. But
that's what made me think of the synergy, because you were saying that you didn't sell mycorrhizae. It was a fungal, and that brought everything together to work together, and that's what I thought.
Yeah, so it's very likely they do synergistic, because below ground ecosystem, or pretty much in the ecosystem, we're not alone, and it's all community, it's all population, is everybody's doing something, everybody's trying to survive. And when it comes to cannabis, it's a simple environment, and it's a disturbed environment, because that's not substrate that it was formed for millions of years. It's a substrate that you made a week before, or a month before. So doesn't have any structure. Everything is artificial. And so anything that you put in there, it's in a chaotic mode, because that's not how they live in nature. And so bacteria and fungi and anything you put in there, they're all in survival mode. Let's eat, let's grow and eat and reproduce, because who knows what these guys are doing up there. We keep getting water, and we keep getting nutrients, and not protozoa coming, and what the hell, I don't know these guys come from, and everybody's on a survival mode. And so that's another way to look at it. Most of the urban landscapes and most of the towns are disturbed environments. Unless you go to some pristine forest, everything is in a chaos mode.
Is it true that, and I know that we've had 1 or 2 other people tell us this before, that there's only one type of mycorrhizae that associates with cannabis? I forget what the first name is, irregularis, or is that not true? Because I know you'll be the person to ask.
Yeah, it's a mycorrhizae fungal species, Rhizophagus irregularis. It used to be called glomus, but there's also glomus interradices, and so on and so on. As I say, what Josh and Leighton are doing is hopefully will pin down if the strains from this particular producer actually do some benefit. We know they colonize, or they will colonize, but that's about it. We don't know if they actually do any benefit unless we measure something. And what are we going to measure? Height, flower numbers, stem diameter, number of, I don't know what you guys measure.
Trichome?
Well, THC and
THC levels, that would be the very fine, because
yield.
Yeah, I mean, I think the THC will be something that will be extremely important for CBDs, because imagine if you find a strain compared to the control that can produce high THC levels versus the control. Wouldn't that be the one of the best goals you want to find
Exactly.
and to stick with that?
Yeah, I mean, obviously that's, I mean, that's the goal. I was wanting to go back to a question before that Steve was asking, and I think you corrected me on and trying to make sure I understand it correctly. So, like when we, there's a practice of adding like barley or different products like that to increase, or even oatmeal to increase the mycorrhizae in quotes.
Yeah, I'm not sure. I'm not sure that is the case because, again, those will be indirect nutrients for whatever microbes are breaking down these compounds that eventually will be taken by the plant. The mycorrhizal fungus is going to feed on basic stuff: phosphorus, some nitrogen, and so on, micronutrients. But the energy source is going to come from the plant. And so
Boom.
the thing
The
is,
metabolism, right?
yeah. So the energy
Boom.
is coming from the roots. And so whatever you're feeding on the substrate is to feed the bacteria, not the mycorrhizal fungi, or a decomposer fungus. They'll break down nutrients, so the nutrients will be available for the plant.
So when I apply, like some of those things to my no-till bed that has, a layer of straw on it, and I see a fungal web,
That's decomposer fungi.
that is not mycorrhizae.
Nope. I can tell you 100%.
That was my point that I was trying to drive home, and I'm
Yeah.
guilty. You can look on my Instagram of me bragging about my mycorrhizae. You can see. So like, I'm just trying to call that out.
Yeah,
That's awesome. That's awesome to solve something like that.
Decomposer fungi do serve the purpose of breaking down nutrients for the plants.
Yeah, I can tell you that
but that's not mycorrhizae. That was my point. Is that we
mycorrhizal
can't
fungi
see it, right?
Yeah, mycorrhizal fungi are invisible unless you stain them.
Say that like 3 more times.
I mean,
So
invisible.
you're saying if you can actually see the mycelium growing, that it's most likely a decomposer?
Very likely, because I'll take it back. I'm thinking mycologists might be listening to me. Okay, here, I'll do it again. So if you see white threads on your substrate, it's a bundle of fungal hyphae making mycelia, right? These threads are, I can assure you, 99.9% are decomposer fungi. Mycorrhizal fungi will not make visible mycelia in the substrate. It's so small that you need to see it under the compound scope. And that's the reason why we clear and stain the roots to actually see the fungal hyphae, because it's almost impossible to see them, especially when they're substrate. Now, when I extract commercial inocula, in the commercial inocula, most of the fungal hyphae are fragmented, and I extract them along with the spores. I can see fungal hyphae, but it's because I extracted it from the substrate, which is clay and other stuff. And I can see, threads of hyphae, and it's just like fine fibers. But if I put a few drops of stain on that liquid, the hyphae is stained immediately, and then I can see it, and then I can quantify it. But I can only do that for commercial inocula because I know that the hyphae are mycorrhizal fungal hyphae. But if they send me substrate with organic matter and a bunch of junk and stuff, you get all kinds of decomposer hyphae. You get brown hyphae, which is not mycorrhizal hyphae, and you got other stuff. And so, and even little pathogens, they're growing in there. So I said, I can't distinguish mycorrhizae fungi from decomposer fungi is just too messy. So I can't tell you anything about, but the spores are stand out, and so we go from there.
Well, you've opened up a few new laboratories here today, because we'll all be experimenting with some of this stuff. I'm sure we'll keep in touch.
Just run first. Write down the question that you want to answer, and that's basically an hypothesis. Hypothesis is you set it up and you say, I'm going to apply this, and it's going to do this. The alternative hypothesis is not going to do that. It's a yes or no.
So it fails or it succeeds.
Yeah, it's a yes or no. And then you set enough replicates. You set everything as the same thing for everybody except the treatment. After that, you got to measure a parameter that is going to give you the answer that you're looking for. Because otherwise, you spend 3 or 4 months and then you don't know what you're going to measure.
I think we've come to measure, like I said, and I just got this, because of the measurements that we've done with LED lighting, that it came down to THC, CBD levels, and yield.
It comes down to total cannabinoids, period.
And then total cannabinoids.
Total cannabinoids and terpenes. Yep.
Also, which is important to me, is how strong it is to combat, pest and disease too. How resistant it is to pest and disease.
Now, one thing you say about lightning, and I don't know if this is possible, but sometimes if you do lightning or the lighting thing,
LED.
If you move the plants around, frequently, that's the best thing. Because if you just let them sit, then you get the edge effect and blah blah blah. Just as long as you say it, so people will know that you're gonna try something. It says, move your plants around frequently, so there's no edge effect.
Right, get different light angles and all for it. And all different parts of the plants get light and all. Yeah. In fact, turn them a quarter to a half a turn every time you water or feed them every 2 or 3 days.
Now, do you guys measure THC levels? So every time you grow something?
Yes, in California and most legal states, you can send off between $40 and $60. In most states now, you can have anything tested that you're growing. So we test pretty much everything, and that helps us. In fact, I've done extensive testing for different lighting, as far as different types of LEDs, different and all that, and I can tell you a whole lot about that. But yes, we do extensive testing every time, and for state anything we sell to the state. Or anything we sell legally in the state, we have to have state testing that gives us all of that data.
But Steve, honestly, Steve, you've probably done more like research and comparison data than most folks. Like this testing most folks are doing is just like a requirement of to pass, fail, or whatever the THC CBD ratios are. It's very simple testing.
Yep. Yeah, so in fact, some of the stuff that we've learned in aquaponics is that aquaponics has higher THCV and CBD levels on average compared to soil controls. But I know Dr. Efren's trying to get going, so thanks. I know you wanna get running. So
if somebody has a question before.
Oh, sure.
Anybody from chat?
Anybody have any other questions for Dr. Efren? Let me scroll up here and see if there's any from earlier.
Well, you're looking. I want to say, Dr. Efren, thank you so much. I wish I would have had an opportunity to take your class, but I'm on the East Coast, and I thoroughly enjoyed. I was riveted to your conversation and style and sense of humor.
Where's the East Coast? We're in the East Coast.
I live in the hellhole swamp in South Carolina.
In the South, and less, and like we like to say, is less than legal land.
But I live in the swamp, and I've got all kinds of you can't stop things from growing here. So I've got all kinds of biological.
Shrek sometimes.
Yeah,
huh?
I feel like putting my boots on for the copperheads already, just thinking about it.
Oh no! That's what we got dogs for. They run through there and run them. There's not a lot of copperheads. snakes. Snakes are they don't want to mess with you. And most people don't know it, but a snake. We'll do this, and then we'll get back to Steve finishing the show. But but most people don't know this. You run across a rattlesnake, it can only strike you about barely over half the length of its body. So if it's a 6-foot snake and you're 4 feet away, you get a 4-foot stick.
Not true.
Yeah, it is true.
No, it's not true. I've bred over 500 venomous snakes. That's not true. I've been bitten by 2 different species of venomous snakes. That's not true.
Well, there's some snakes, but in general, when you run across rattlesnakes and copperheads and stuff like that, they can only strike you.
3/4 of the body. Like,
all right, well, see, you want to err on.
Anyways, I can tell you firsthand. I used to deal with shit that was highly venomous. That's one thing
I open a new.
Yeah.
Well, I worked with a forester and cleared roads in the swamp. So,
I open a new can of snakes, right?
But see, I'm sure Steve's right too. Let's just leave it at that. He's right too.
We had a guy say much love from New Zealand. You have a big fan in New Zealand.
Any other question?
We don't have any other questions from chat. Does anyone from the panel have any other questions?
No,
not that I can think of.
You want to tell people how to find you and how to find your company? And I have your information also in the description of the video as well.
Yeah, mycoroots, mycoroots mycoroots at comcast.net or mycoroots.com. It comes out quickly. So yeah, I'm consulting. I'm all over the place. I say I'm going to be traveling quite a bit. New York and.
Can I add in if you're on the commercial level, and you want to buy legitimate mycorrhizae spores, that folks could contact you in order to source that.
Yeah,
I know you don't grow them, but
actually, I don't sell products.
You don't sell products, but I guess in my case, I understood that you tested it before and you.
Commercially, I'm not supposed to recommend, but publicly.
So don't get in trouble.
Okay. Yeah. So because most companies they do pretty good stuff. But I just like anybody. When you go to the supermarket, you have 5 different kinds of milk to choose from, and you go for the one that you like. And so
right. But maybe if folks wanted to get real serious, they could send their product that they're going to purchase to you.
Oh yeah,
test it.
Oh yeah,
before they use it, and you could say, "Hey, this is legit, and this is full of spores."
Gotcha. Usually, a lot of the assessments are obviously confidential because I don't, you will not believe the history of this industry. A lot of times, people want to talk negatively about other companies, and so they says, okay, I'm gonna send you a product, and then they get pissed off at that company. They want their money back, and and then I get in the middle of it, I'm here to support the industry. I'm here to support the clients that want to use them, and so I'm a friendly guy, and I'm not here to cause any problems.
Hard feelings. We understand that, Doctor Efren, because Steve's the same way. Steve always buys the product. He never lets anybody give him the product. So then when he reviews the product, he can do it with and hold his head high because he bought it. He didn't get it given to him by the company, so therefore his is. But in your case, I can still understand you have to deal with all these different labs and producers.
And the thing is,
be cool.
As far as I know, there's a couple other labs that they do some assessments, but I never been to those labs. I don't know them personally, but I know there's a couple other labs But I mean, I'm one man orchestra. I don't have technicians. I do the whole thing myself. Recently, I've been overwhelming with some stuff, so I'm getting my son to help me here, and to do some of the technical stuff. But I like to take care of the whole thing myself. That's why I stand by what I do, and I privately you can ask me what do you think of this product, but there's always want to make sure off the record or confidential, blah blah, the whole thing, just to make sure nobody gets hurt. I'm not here to put down any company. But yeah, I test products, and just by doing the counts, make sure things are there. Sometimes there's more than what they say. Sometimes they're lower what they say. At least based on my methodology.
And roots. Like I guess I wasn't trying to like talk about dogging a product, just about the like.
And the roots.
You could help folks figure that out. That's part of what you do.
Yeah, exactly. You put your product, you let them grow for at least 3 months if you can, and then we can check the roots and to see if they actually work or not. Because if you apply something, you're assuming it's working, and it's not working. Maybe you want to change product or don't use it at all.
Oh, perfect. Great job, Josh. You bringing it around to what that's what you meant. I understand. Yeah. So that's what your business is that you will test roots.
Yeah, I do it roots assessments. Yes.
That's okay. See, I don't think we really hit that.
That's what I was trying to hit at. Like,
yeah, thank you, Josh.
You do root
test the roots.
Yeah,
test the product before you apply it and tell you if it actually has the spores.
Yeah,
he can help you all along the process of working with whatever product. And I wasn't asking him to call out any product. I was just trying to clarify that that's what you do is you and that analysis.
Yeah, thank you.
Or the other side is you apply the product and send the roots to you, and you look at the roots.
Thank you, Josh.
After you've used the product.
Yeah, I mean that's what I do. Let's call mycorrhizae assessments based on the roots. Either from the native communities or something that you're growing that you apply. So you send me a handful of roots, and they have to be from that individual that you're testing, and then we clear them, and I stain them and look under the scope and tell you percentage colonization, and then you can decide, did my product work or not, or whatever I'm doing is working. Because if I don't find anything on those samples, that product is definitely not working. It's just the bottom line.
Then we got to the root of how to get your business going.
There's no, I mean, that's why we use colonization as a sign of a healthy soil.
And it takes 90 days for colonization. That's why I was asking very specific questions about the way we grow.
Because that's my
something about that 90-day thing. See, my plants are never done in 90 days. So if I started doing this at the beginning of my grows take 120 days generally.
Well, I mean, even 120 days even better. But
that's 90 days. I don't know. Yeah.
No, I mean you can do the assessment at 90 days, and usually you can do it at 120 when you're done. So it will be fine. And it's a range time, but there's some species that some people test it after 8 weeks. Like after 60 days.
Oh, so that's all you're saying is that the testing is invaluable up until 90 days.
Yeah, because some colleagues, some paper says, yeah, we did the assessment after 8 weeks. To me personally, it's just the beginning. To me, that doesn't mean that some strains can work a lot earlier than others. But I mean, you can test them every month if you want to, But obviously, it's going to be budget, and you don't want to damage your plants and so on. So I rather wait, 3 months at least. Commercial people even wait 4 months, and then do assessment to see how their production came up. So you can choose between 90 days and 120 days if possible. But you can have a crop that is growing, for 6 months, and you can assess them at the end of the year at the end of the crop. That's fine too.
Right. Right. Because it doesn't have to be cannabis all the time, but we did get
no, it's just snap. It's just the colonization, pretty much in any data, any lab data is a snapshot of in space and time of what you're trying to measure. If you do it a week before or a week later, you may have different numbers.
Oh yeah, for sure.
So
I mean,
So real quickly, I thought of a question. Is there anything that people can be doing to try and capture their local native mycorrhizal fungi?
we touched on that earlier.
you want to go back to the secrets, huh?
Well, see, that's no. I'm not going to hijack. I had but I was going to say. Well, I'm going to say because it's funny. I'll let you address that. But I was just funny because when you were saying, you were about secrets, and I knew better than to ask you, well, how do you dye the roots so you could see it, and then
yeah.
If you want to tell us the secret, then I'm.
Oh yeah, well that's not a secret. We use Trypan Blue. I use Trypan Blue, but there are all kinds of things. You can use aniline blue. I mean, they're all nasty chemicals. At some point, somebody in Canada try black ink. Regular ink, Pelican ink. They try all kinds of inks, and they did it mix it with vinegar, and so they're trying to do some kind of a less carcinogenic.
So it's still experimental too.
No, I mean it works, but it doesn't. The stain is kind of you use it one time and you're pretty much done. And when I use Trypan Blue, as long as you use gloves and air it, that's not an issue. I've been doing that for 30 years. You clear them with KOH, this potassium hydroxide at 10%. You can get that on Amazon. You clear the roots, and then you put them. Yeah, and then you put you dump them. You clear them for 24 hours, room temperature, and then you dump the KOH. You put acid solution, 1% of hydrochloric acid. You can replace it with vinegar if that matters. It's just an acid. For 30 minutes, you dump the acid, and then you put the solution of the stain, which is Trypan Blue and lactoglycerol, as lactic acid, glycerol, and water, 1 part each. And then you put 0.5 gram of the Trypan Blue. The Trypan Blue is very potent, so 0.5 grams is 500 milligrams. So if you mix that at a 1 liter of this solution of water, water, lactic acid, and glycerol, you make it at 0.05%. And so that solution can be reused. You use it and use it, and it only takes a few hours. In a water bath, you put those roots in the vial, in a little seed vial. So they're contained, and you can do a bunch at a time. And then after that, you dump the stain, and then you just add water, and your roots are ready to look at under the scope.
Wow, thank you. That's awesome.
Once you send me roots, I send you the method back. I can tell you what the method I use.
Right. So now back to the secrets. I thought that Brain Grow asked you the same question, but was that a different question than you were asked earlier as far as the
No, I cut out a couple of times, so I may have missed it, and I apologize if I asked the same question twice.
Well, we talked about how to find the saltiest in the show, about you go to a place that you want a fungi that would resist.
Okay, gotcha. Okay, I was paying attention there. I don't know. It's been a pretty
then you
long
go
first.
to the tolerant species. You go to the desert. You go to whatever feature you're trying to select. You go to where it's happening naturally. And then
so you dig up, according to Doctor, you dig up the plants that you find that live on the first plants that live on the edge of the salty area to be salt resistant.
It's either colonized first.
Yeah, and then send them to him, the roots, and he'll tell you if they've got.
Yeah, and then that soil is very likely the habitat for this type of fungi. And then from there, you start, isolating them. It takes a few hocus pocus. And then
Well, I
Thanks a
can
lot.
see that you need your cookies and milk. I think we should let you.
Yeah.
Somebody needs to wind you up, or something.
I think Steve is getting cold now. He's wearing a jacket.
Yeah, you were awesome though. I'll tell you what, I was riveted to your explanation of things tonight, and I understood most of it too. So that was really cool.
Well, this is Steve's idea because he wants to warm up for the conference in Humboldt. And
Well, Steve's awesome, man. He gets all you great guys to come and share your knowledge with us, and we badger you for a while and have fun.
I think my talk is on Friday.
Yes, sir.
That's tomorrow.
The 16th. No, the 16th. In Redway, California, which is in the Humboldt County. I'll be flying in on Thursday. And unfortunately, I'm flying back Saturday.
I'll see you Friday night then.
Yeah, they tell me that there's only one traffic light.
It's a small little area we're going to. I think we're all hanging out pretty close quarters, so it'll be fun.
Well, I tell you what, you guys are really doing something. I love listening to all of you guys, and I envy you so much that you're out there being able to go.
We got to have you out
every week
for one of these, dude.
All right. Well, thank you guys. I say half an hour. I think I lasted 2 hours. It's all your fault.
Yeah,
Well,
you're
thank
awesome.
You thank you for your time, and we much appreciate it. Thanks again.
All right, guys.
Thank you, Doctor.
Now, how do I do it? Do I click and
Oh, you
close?
just close. Yeah, just close your window. Thank you.
All right, guys.
Cheers.
Stay in
Good night.
touch. Stay in